Immune hemolytic anemias are flagged clinically by a hemolytic anemia that develops recently, prompting the clinician to suspect an immune mechanism. The most common form is warm antibody autoimmune hemolytic anemia, which is driven by IgG autoantibodies that attach to red cell surface antigens, most often Rh blood group antigens. These IgG antibodies are called warm because they function optimally at body temperature, 37 degrees Celsius. This condition is frequently secondary to another underlying disease, including systemic lupus erythematosus, Hodgkin disease, or non-Hodgkin lymphomas. Clinically, this anemia presents with the general features of any hemolytic process, but also with spherocytosis, which here develops not from a genetic membrane problem, but from progressive loss of membrane material as antibody-coated red cells repeatedly pass through the spleen and get partially consumed. The most diagnostically critical finding is a positive direct Coombs test, also known as the direct antiglobulin test, or DAT. This test detects IgG autoantibody that is bound directly to the red cell surface, confirming the immune origin of the hemolysis. Cold agglutinin disease is the second major immune hemolytic anemia and is driven by IgM antibodies that work best at temperatures below 30 degrees Celsius. The chronic form, also called cold agglutinin syndrome, is characterized by hemolysis and red cell clumping in body parts exposed to cold temperatures, with associated Raynaud phenomenon. It often connects to lymphoid neoplasms, and the responsible antibody typically targets the blood group antigen called I. The acute form is transient, usually following an infection. Anti-I antibodies emerge after Epstein-Barr virus infection, infectious mononucleosis, while anti-I antibodies appear with Mycoplasma pneumoniae infection, and the finding of cold agglutinins can actually help make the mycoplasma diagnosis. Warm autoantibodies are usually IgG1, optimally reactive at 37 degrees Fahrenheit, and positive for IgG or IgG plus C3 on direct antiglobulin test, DAT, target mainly Rh blood group antigens. Cold autoantibodies are mostly monoclonal IgM that react below 30 degrees Celsius, optimum 4 degrees Celsius, and activate complement, with the DAT usually C3 positive and negative for IgG. Common targets include I or PR antigens. Warm types present with anemia, jaundice, splenomegaly, and evidence of hemolysis. Cold types can cause acrocyanosis, hemoglobinuria upon cold exposure, and symptoms are often exacerbated by cold. Laboratory findings include anemia, reticulocytosis, increased indirect bilirubin, low haptoglobin, and characteristic DAT patterns, IgG plus for warm, C3 plus for cold. Cold agglutinin titers superior to 1 to 64, supports cold type. Warm AIHA typically responds to corticosteroids as first line, with rituximab or splenectomy for refractory cases. Cold types often require cold avoidance, rituximab, and in severe cases, targeted therapies such as anti-complement agents.